Good morning and welcome to the CRISPR and NGS Symposium. My name is Rafi Gatzel and I'm the Assistant Managing Director at Syntezza Bioscience. Our company is pleased to bring you today's symposium. I would like to thank the Sackler Interdepartmental Core Facility, Sackler Faculty of Medicine, and the Tel Aviv University for hosting the symposium. I'd also like to thank Sippy Borstein, Karen Abram, Noam Shomron, Karnit Levy, and Gilad Mess, you just heard. I would also like to thank Integrated DNA Technologies and Syntezza Bioscience for helping to sponsor today's events, and all of our customers who are here. The large crowd we have here today is a testimony to the interest in CRISPR and in next-generation sequencing, which are two of the technologies that are at the forefront of biotechnology research today. The ability to efficiently sequence on a genomic scale and to edit the genomes of living cells are cutting-edge technologies that we're only beginning to reap the benefits of. Each of today's speakers is an individual with whom we at Syntezza have had the privilege of working. We are honored to play a role in advancing the role of CRISPR and NGS in the toolbox of molecular technologies here in Israel. It is my pleasure to introduce our first speaker of today, Professor Ayal Hendel. Ayal Hendel is currently a principal investigator and senior lecturer at the Mina and Everett Goodman Faculty of Life Sciences at the Bar-Ilan University. Ayal's research focuses on developing genome editing and CRISPR technology as a method of gene therapy for genetic disease and of the blood and of the immune system. Ayal received his BA with high honors from the University of Tel Aviv, from Hebrew University of Jerusalem in biology, and his master's in science and PhD from the Weizmann Institute of Science. Ayal recently returned from a 5-year postdoc at Stanford University, where he was the first to demonstrate that chemical alterations to synthesize CRISPR single-guide RNAs enhance genome editing efficiency in human primary T cells and hematopoietic stem and progenitor cells. In addition, in a recent study, he was part of the team that achieved therapeutically relevant genome editing frequencies in human hematopoietic stem and progenitor cells. The title of Ayal's talk for us today is CRISPR Genome Editing of Human Hematopoietic Stem and Progenitor Cells, the Promises and Challenges.
Can everybody hear me in the back? Okay, thank you. So thank you very much for the nice introduction, and I'm very excited to be here today and to tell you about my work. And what I'm going to do today is I'm going to tell you about my research, mainly the one that I did at Stanford, but I also keep the last 10 minutes to answer questions because I see that most of the people in the audience are actually users that use their technology for the research, mainly for doing genetics, but maybe some of you also are planning to use it for translational research. So the main focus of my research is actually developing genome editing strategies for genetic disorders of the blood and the immune system. And what you can see over here in this picture, here you can see a picture of sickle cell that has sickle font mutation of codon 6, A to a T, leading to the disease sickle cell anemia. And over here, you can see the picture of David Vetter, who back in the battle days was kept in this bubble, as you can see here behind this sterile environment for almost 14 years because he was born with mutation in the gene that you can see over here, interleukin-2 receptor of common gamma chain, or in short, R2RG, leading to inability to develop T cells and NK cells, namely inability to develop functional immune system. And for this reason, he was kept in this battle for almost 14 years. And these were early 80s, the first days of the treatment of this disease, which was allogenic bone marrow transplant. And immediately after the procedure, the transplant, he actually died from complication of this procedure. So pretty much what we can use as a therapy for these two devastating disorders, which are very devastating and life-threatening, is allogenic bone marrow transplant. And I'm not a clinician, but just to briefly explain what is allogenic bone marrow transplant, we take hematopoietic stem cells from healthy donors that do not have the mutation in the disease-causing gene, and we give these cells to the patient. And thereby we can create normal hematopoietic system. We can generate normal blood cells and immune cells. And by taking this approach, we can actually cure the disease. This is the only curative therapy for these type of disorders. But there are some challenges with this strategy. First, less than 25% of the patients actually have matched donor. And in cases where we cannot find matched donor, one possible outcome of the allogeneic bone marrow transplant is what we call graft versus host disease, which occurs when the donor cells that come from the donor attack the patient cells because they appear foreign to them. Another possible outcome is graft rejection, when now the patient cells attack the donor cells because, again, they appear foreign to them. So to overcome these problems, what we need to use are drugs and medications that pretty much suppress the immune system and try to solve this problem. But if you suppress the immune system, there are other problems that can appear now. So an alternative way to the allogeneic bone marrow transplant is actually to take the patient's own cells to correct the disease-causing mutation and give the cells back to the patient. So how can we do it? The way that our lab and many other labs around the world are working on it is through the concept of genome editing. And I believe that many of you saw this slide before, how genome editing is actually working, but since this is really the core of the technology, I'm going to explain step by step. So the first step in this genome editing technology, which pretty much allows you to correct the disease-causing mutation, is the introduction of the site-specific double-strand break in the chromosome. And once we generate this site-specific double-strand break by engineered nuclease, and there are a few different platforms in engineered nuclease that can induce this break, now the cell has to repair this break. So it's quite awkward because we are correcting something by first breaking the chromosome. So now, once we generated this double-strand break, the cell has to repair this break by its own DNA repair machinery. And this can be done by two different pathways. Either the non-homologous end joining pathway, or in short, the NHJ, or the homology-directed repair pathway, the HDR. So let's start with this pathway, the NHJ. So what is happening is that you have a repair machinery that can take the two sides of the break and ligate them together, bring them together and to seal the break. And there are many proteins that are involved in this process, and actually, most of the time the repair process is error-free, namely, there is a break, and the break is being repaired in error-free manner. But if you repair the break in an error-free manner, then the nuclease, this platform that generates the break, can introduce the break again. And after a few cycles of generating these breaks, eventually there are exonucleases or polymerases that either chew back or insert a few nucleotides to generate short insertion and deletions, what we call indels. And here the desired outcome, both for therapy but also for genetics, is gene disruption. So if we think about therapy, for example, we think about HIV, we can disrupt the receptor that the virus is using to enter the cells. So we can take this strategy to cure HIV. And if we think about genetics, we can, by taking this approach, knock out genes. So this is the NHJ pathway. The second pathway, the HDR, the homology-directed repair pathway, if we provide the cells with a synthetic piece of DNA that we can pretty much introduce to the cells, for example, by plasmid or using same viruses, then the cell can use this repair template in order to repair the break by the homologous recombination. And the structure of this repair template is arms of homology, area with homology to the two sides of the break. This area has homology to this part of the break and this to the other part. And in between, we now can put the precise change that we want to introduce into the genome. And here the desired outcome is either gene correction for therapy, gene editing if we want to study the function of specific nucleotide, gene labeling, or gene addition. Now currently there are three main families of engineered nucleases. And the engineered nuclease, just to go back to the previous slide, are again the platform that introduced the break. So the first platform is zinc finger nucleases. nucleases, ZFNs. The second are the transcription activator-like effector nucleases, or in short, TALENs. And the last one is what I call RNA-guided engineered nucleases, which you probably know by the name of CRISPR. Now, all these platforms, you see that in terms of their structure, they are very different, but in terms of their mechanism, they are very similar. All of them, they have two parts. One part that can bind the DNA, for example here, and we can engineer this part to pretty much recognize almost any sequence that we want. And the second part, the part in the middle over here, is the nuclease, the one that can introduce the site-specific double-strand break. So in this platform, the TAL domain is the domain that we can engineer to recognize the sequence in the genome. And the fourth one is the nuclease that can introduce the break. And over here, when two talents dimerize, when the fourth one, these two talents is dimerizing, then now the fourth one can be active and introduce a double-strand break. Now here you can see example of the CRISPR system, where in nature the main purpose of this system is actually adaptive immune system of bacteria against bacteriophages, the viruses that attack the bacteria, or against foreign DNAs like plasmids. And over here in the engineered CRISPR system, you can see that the single guide RNA, the RNA part, is giving the addresses targeting the Cas9, which is behind the protein, which is the endonuclease, to introduce the site-specific double-strand break. Now, when pretty much new set of engineered nuclease is being developed, and I heard before my lecture that a few of you are trying to work with this platform these days, pretty much you need to screen to find the nuclease with best activity, and many times for different applications and different cell types, you need to measure the genome editing activity in order to find the best nuclease. And this thing has been previously been made by a variety of assays, including enzymatic-based assay, reporter-based assay, and also a second-generation sequencing technology. But each of these assays that I just mentioned has their own utilities, but they also have major limitations, including insufficient sensitivity, the inability to measure simultaneously the NHEJ and the HDR outcome. They need to use a reporter cell line, and if you use second-generation sequencing technology, the read length limitation of this technology. So I decided to use a new technology to develop a method that can simultaneously measure the two outcomes, the NHEJ and HDR. And for this reason, I decided to work with long-read sequencing provided by a company called PacBio. So just to briefly summarize what I learned from using this technology, first I learned that pretty much if you deliver more nuclease to the cells, you can get more NHEJ and HDR, the two different outcomes. And this graph is showing that. So in this experiment that you can see over here, I titrated up the amount of nuclease. In this experiment, I used talents, and kept the amount of donor DNA in the cell constant. And what you can see over here in the red and the blue bar, that the NHEJ and HDR frequency rose when I titrated up the amount of, in this case, talents in this experiment. So pretty much, if you want to get more NHEJ and HDR, you need to deliver more of the nuclease into the cells. Then we learned that more donor shifts to more HDR. Sometimes you want to, let's say, correct a disease-causing mutation, or you want to change a specific nucleotide into the genome. And for this reason, you need a homologous recombination pathway. So what is the best way to achieve that? So in this experiment, what I did is I kept the amount of nuclease, again, talents, constant in this experiment, and titrated up the amount of donor DNA. And what I saw over here is shift in the ratio of homologous recombination to NHEJ until a point that I was able to get more homologous recombination than NHEJ in this experiment. And the last thing is we were able to see that arms of homology, this area of homology on the two sides of the break, which were around 400 base pairs, gave us the maximal rate of homologous recombination. So as I told you, the structure of this repair template is in blue. You can see the exact modification that you want to introduce to the genome. And this is example of a repair template. And then you need to have this homology to the two sides of the break around this chain. And homology of 400 base pairs gave the maximal frequencies of homologous recombination. And this was done in cells in a cell line called K562, but we also can see these days with other cell lines. There is no need to have homology longer than that. So far I showed you many data coming from CRISPR, but we also wanted to see, okay, this was almost 2 years ago, and everybody started to work with the CRISPR system. Let's see what is going on with the CRISPR system compared to the TALEN system. And we tested CRISPR and TALENs that target two different sites, two different loci, R2RG and the HBB, the one that are related to the two disorders that I showed you on the first slide. And what we saw that in these two cases, we got better activity with the CRISPR system, both when we measured the NHEJ and the HDR frequencies. So many times, if you just want to get a very fast active nucleus, the best way to get it is to start work with the CRISPR, because even if you test 1, 2, or 3 different CRISPRs, there is high chance that you will find a very active CRISPR. If you are going to test more TALENs nucleases, eventually you will find one that can be as active as the CRISPR system, but it will take you more time. But what was very interesting in this experiment is, despite the fact that when we measured the NHEJ frequency in yellow in these two sites, and the CRISPR system gave very high activity, when we measured the homologous recombination frequencies across these two sites, we got pretty high activity for the R2RG site with the CRISPR system, but much lower activity with the CRISPR system. So this really showed us that genome editing outcomes can be affected both by the platform that you are using, namely the CRISPR compared to the TALEN, but also by the site that you're trying to target. And we still don't understand what are the parameters that are related to these differences between these sites, whether it's the sequence composition, whether these are epigenetic parameters of these sites, but you definitely can see that genome editing can be affected by more than one parameter. We also were able to see this very interesting observation. Since we used long read sequencing, we were able to actually identify pieces of DNA that were captured inside the break. So I told you the first step in genome editing is generating the break. And then you want, let's say, to correct the disease-causing mutation. But what we saw is that pieces of DNA that were part of the genome editing reaction were captured in this break. And these pieces came from the plasmid that express either the TALEN or the CRISPR or the donor DNA. So this really showed us that in order to minimize this toxicity or off-target outcome, you want to minimize, to take out as much as you can the DNA from this reaction. Now, in order to use this technology for gene therapy application, what you really need to do is to engineer the primary cells, the cells that are relevant to the clinic. And in our case, we speak about the hematopoietic stem cells. So when we went to test the CRISPR system in the hematopoietic stem cells, we saw very interesting things. So when we tested the CRISPR system compared to the TALEN system, as I showed you in the K562 cells, and the CRISPR and the TALEN were expressed from a plasmid, the CRISPR system worked better than the TALEN system. But when we switched to the hematopoietic stem and progenitor cells, we got very low activity with the CRISPR system. And what you can see over here is actually an error bar. So we were able to measure 1% of homologous recombination with the TALEN system, expressed from a plasmid in the primary cells, and very low activity with the CRISPR system. So this really told us that something about, in this case, the formulation that we were using was not working in the hematopoietic stem cells. And at this point, I was actually in a meeting at Stanford, listening to a scientist from Agilent Labs, demonstrating their ability to pretty much chemically synthesize the single guide RNA, the same way that people are synthesizing, let's say, the siRNA if they bind siRNA. But the challenge with single guide RNA, the RNA part of the CRISPR system, is that it is much longer compared to siRNA. It's around 100 nucleotides. And we also knew that delivering thing as a plasmid to primary cells is very toxic because we had a few gene expression studies that show us that if you deliver DNA, let's say even plasmid, into primary cells, you get pretty toxic response. So immediately after this meeting, I established a collaboration with Agilent Lab saying maybe we can chemically synthesize the single guide RNA and deliver it directly into the cells to rescue the activity in the primary cells. And one main advantage of the ability to synthesize the guide RNA is the ability to incorporate modification into the molecule. So pretty much we incorporated nucleotide modification at both the 5' and 3' terminal ends of the single guide RNA. And this part is the single guide RNA of the CRISPR system. And the modifications were chosen based on the resistance to exonuclease activity. So if you deliver, everybody are familiar with the fact that RNA is very sensitive. There are exonucleases that can chew the molecule. But we decided to put actually modification at the ends of the molecules to block this exonuclease activity. Now, the modification that we tested, you can see over here. And this was interesting time during my postdoc. I've never believed before that I'm going to go back to my notebooks in organic chemistry to review this material, but this was the first time that I went back to review and learn more about this type of modification on the RNA molecule. So what you can see over here is what we call a vanilla RNA, unmodified RNA. This is the structure of RNA. And we tested 3 different levels of modification, what we call M, MS, and MSP. And pretty much in the end, what you see is 2' OMP modification on the sugar group. Over here, you can see the same modification plus a tire group on the phosphate backbone. And over here, on top of these two, you see a acetate group on the other side of the phosphate backbone. So we decided to test these level of modifications, and at that point, there was quite a lot of knowledge of how to modify RNAs that came from the world of siRNA and antisense oligo, but there wasn't any knowledge whether you can modify the RNA and still keep it functional inside the cells. So the first experiment was to start again with cell line, K562 cells, to deliver these synthetic RNAs into the cells and see whether we can modify the RNA and still keep the molecule active. And what you can see over here in the gray bar, activity of CRISPR expressed from a plasmid. We got very high indel activity, NHEJ activity, when the CRISPR was expressed from a plasmid. But when we switched to the MS and MSP modification, we got SI or higher activity inside the cells. But just to demonstrate that these modifications are actually important inside the cells, we decided to use small amount of these synthetic guide RNAs. These synthetic guide RNAs were delivered into the K562 cells by electroporation. And what you can see over here in the light bars is delivering of only 1 microgram of these synthetic guide RNAs into the cells. And you can see is that if you deliver only 1 microgram, unmodified RNA is giving us 2% of activity, M is giving 14, MS is giving 68, and MSP is giving 76% of activity, more than 32-fold higher than the same amount of the unmodified RNA. And pretty much we got the same result for the HBB and CCR5 genes. So I told you that delivering DNA into primary cells is pretty toxic, and we wanted to see whether we can switch to what we call all RNA formulation, namely both the guide RNA is going to be delivered as synthetic guide RNA, but also the Cas9 delivered directly as mRNA. And there is actually a big trend these days to delivering more and more things directly as mRNA into the cell. So by titrating up the amount of Cas9 mRNA and guide RNA, we were able to achieve very high NHEJ frequencies in these cells. So in this reaction, there is actually no DNA when we deliver the CRISPR system. And in this case, we even switched to the next generation of CRISPR. We deliver the guide RNA, the synthetic guide RNA, and the Cas9 directly as a protein. So what you can do is you can take the RNA and complex it with purified Cas9 protein, and you can deliver it into the cells by electroporation. And again, you can see that with the MS modification, we were able to get very high activity. And the advantage of using Cas9 as a protein is that there is very small amount of nucleic acid in this reaction, which is much less toxic to primary cells. So of course now the important thing is to see whether we can get high editing efficiency in these cells that are relevant to the clinic, the hematopoietic stem and progenitor cells. And what you can see over here for these two sites, alpha G and HBB, is that if you deliver the CRISPR as a plasmid, you can't see any activity. But if you now complement the system, still the Cas9 is expressed for the plasmid, and you deliver the guide RNA with the modification, now you can rescue the activity. And if you now also deliver the Cas9 as mRNA, you can get pretty high activity in the hematopoietic stem and progenitor cells. And also we got the same high activity in primary human T cells. And these two types of cells are very important to the clinic. The hematopoietic stem cells are important for curing monogenic disorders like what I just showed you, sickle cell anemia and severe combined immune deficiency. But if you think about T cells, this is really important for cancer immunotherapy because these days people are looking into modified T cells in order to add them specific receptor that will be able to recognize cancer cells. And one way to do it is by using the CRISPR system. And now we can achieve also very high level of modifications in the primary T cells. Now we wanted to see if we can even increase more the activity of gene disruption, and we decided to test what we call dual single guide RNA approach, namely using two guides that cut next to each other. And here you can see the distance between these two guides was 205 base pairs. And by taking this approach, delivering simultaneously two guides into the cells, we were able to increase the gene disruption frequencies from 70% to 90% in the T cells, and from around 20% to 40% inside the hematopoietic stem cells. And if you take this approach, you have to remember that you're going to get pretty much very complex set of events, because what can happen in the main event over here is just deleting this piece between the two guides, but you can also get some other type of events. This piece of DNA can go out and come back in the reverse orientation, or you can get just small insertion deletion at both sides or only in one of them. And the only way to characterize these outcomes is by DNA sequencing. Now I'm showing you this slide just to hammer the point that the all-RNA CRISPR system lowering the toxicity in the primary cells, and in this case primary T cells. And what you can see over here is that the DNA is increasing cell death and decreasing cell proliferation with compared to all-RNA system. Now this slide is a little bit complicated, so try to stay with me, but this slide is really showing the importance of using the Cas9 as a protein. What you can see over here you can see the indel frequencies, the NHEJ outcome, when you measure genome editing at on and several off-target sites, and you use unmodified which compared to the MS-modified guide RNAs. And in the colors, what you see is yellow is Cas9 expressed from a plasmid, green is Cas9 expressed from the mRNA, and red is Cas9 delivered directly as a protein into the cell. So we have indel frequencies, we have on-target and few off-target sites, unmodified and modified RNAs, and three different ways to deliver the Cas9. And what you can see over here very nicely is that in all cases we got higher activity with the modifications, both at the on-target but also at off-target sites. But if you look over here at these off-target sites, when you deliver the Cas9 as a protein, we got the lowest off-target activity. So this really is showing us that there is a sweet spot for nuclease activity. You want to have enough nuclease inside the cells to get high on-target activity, but you don't want to have this nuclease for a long time inside the cells, because this is going to induce also off-target activity. And what you can see over here in this illustration, the red line is representing Cas9 expressed on a plasmid. The blue line is Cas9 delivered as a protein, and the black line is what you want to get in order to achieve optimal genome editing outcome. And I call it actually the hit and run away mode of action. If you think about siRNA and antisense oligo, what you want to have over there is a constitutive activity. You want to sustain the activity in order to see downregulation of the specific gene. But with the CRISPR system, because we are modifying the genome, you want to induce this change, but you don't want to have these scissors inside the cells, because if you are going to have the scissors for a long time, this is going to induce also off-target activity. So now, what I showed you so far is really how to induce insertion or deletion, but in order to induce a specific change by homologous recombination or to correct a disease-causing mutation, what you need is to deliver the repair template. And for a long time, we tried to deliver this repair template by using plasmid. But I just showed you that using plasmid is pretty toxic to the cells. So more or less at the same time that I published this paper with modified RNA, a few groups from the United States showed that they can take other platforms, not the CRISPR system, and actually use a different way to deliver the donor DNA into the cells. And what they use is recombinant AAV6. This is a virus, an engineered virus, as a way to deliver the DNA, the donor DNA into the cells. So this is the genome of recombinant AAV. And when we saw this publication, we immediately went to test that. And what you can see is that if you deliver the CRISPR system with this modified RNA and the donor DNA you deliver with recombinant AAV6, now you can get pretty high activity of homologous recombination inside the cells, whether you are using Cas9 delivered mRNA or as a protein. So just to summarize everything that I showed you today, chemically synthesized single guide RNA significantly enhance genome editing in primary T cells and hematopoietic stem cells. The whole RNA and RNP, this is the part where you deliver the Cas9 as a protein, lower the toxicity in primary cells. Modified single guide RNA holds the potential for further optimization of activity, but also specificity by using different modifications at different sites. The chemically synthesized single guide RNA and recombinant AAV6 induce high homologous recombination frequencies in hematopoietic stem and progenitor cells. And finally, this technology offers robust and scalable production of single guide RNA for therapeutic applications. So really what I want to do right now is to take the next few minutes to tell you about what I think are the challenges of this field and the thing that I'm going to develop in my own lab. So as I told you, my focus is to develop genome editing strategies for genetic disorders of the blood and the immune system. And in order to develop these strategies, what I believe is important to do is to study the link between genome editing and DNA repair, the link between genome editing and chromatin, and to continue to build the synthetic toolset for genome editing. So the first thing is studying the link between. So the first thing is to study the link between genome editing and DNA repair. Here's the specific question that I would like to address: is what are the genes that are associated with HDR and NHEJ mediated genome editing? And it's very interesting, despite the wide interest in genome editing and a lot of interest in DNA repair, we still don't understand what are the repair factors, the genes that are involved in genome editing. And I believe that by finding these factors, we will be able to understand why in specific cell types, let's say the cells that are not dividing, we get much lower genome editing activity compared to the other cells, and we can also make this process much more specific and accurate. The second direction is the link between genome editing and chromatin structure. And here, I would like to ask the question, what is the relationship between chromatin structure and genome editing outcome? And if you think for a second where chromatin structure can affect genome editing, we can think at least on three different levels. First, the binding of these nucleases to the DNA, the efficiency of binding of the CRISPR to the DNA. The second is actually the choice between the different DNA repair pathways, whether it's NHEJ or HDR. And the third level is actually events of genomic instability, such as chromosomal translocation, that can also be affected by a chromatin structure. And here again, I believe that once we learn this information, we will be able to make this platform more active and more precise for many different applications, not only genetics, but also for therapeutic applications. And the last thing is building a synthetic tool set for genome editing. So what I showed you here is that I used only 3 different levels of modifications on the RNA. But actually, by being able to incorporate this modification into the DNA, we pretty much open a whole new language, because it's not the language of the 4 nucleotides. You can put these modifications on the bases, on the sugar, and on the phosphate backbone that can also change the specificity of the RNA. And here, the big challenge is to tweak, to change the binding in such a way that we won't affect the on-target activity, but by putting this modification, we will be able to eliminate activity at off-target sites. And that's because many times the activity at off-target sites is happening because there are mismatches in these off-target sites. And by putting this modification, we can design a way to eliminate the off-target activity and by the same time keeping very high activity in the on-target sites. So right now we have a very good outline how to make autologous gene-corrected hematopoietic stem and progenitor cells. We need to harvest the hematopoietic stem cells and progenitor cells from the patient. We need to correct the disease-causing mutation and then give these cells back to the patient. And the goal of my lab is actually to make this part more efficient, safe, and specific, mainly for therapeutic application. So with that, I would be happy to end and thank my collaborators. I also would like to thank my colleagues from Stanford and the scientists from Agilent Lab that introduced me to the world of modified RNAs. And we are also, since I'm just establishing my own lab, we are looking for a highly motivated experience to join us. So I would be happy to answer any questions if you have any. Thank you. And feel free to ask even if you have any technical questions.
There was a slide that was showing the difference between modified and unmodified with Cas9, plasmid, protein, and mRNAs. And you showed there was the least off-target when Cas9 was a protein, but it was still, all of it was more off-target with modified RNA than with unmodified. So are you looking into that? Is that something that you can think of a mechanism, or does that change between cell lines and cell types? Maybe it relates to what you just said now, how you're building a language with these modifications.
So the question was, dealing with the Cas9 protein, when you use the modification, you still see off-target activity, but you don't see it with the unmodified RNAs. And what is happening with this system is you actually increase the activity, whether it's in on-target or off-target sites. So first, in order to be able to get therapeutic levels, you need to increase the activity, but then also increase the off-target activity. And now we are working on the next level of modification that we will be able to decrease this even more. So there are a few ways to go around it. One is first to try to screen more guide RNAs that eventually won't have activity at off-target sites. The main reason for activity at off-target sites is actually you have a site in the genome with only one mismatch that generates this off-target site. This one mismatch is still enabling for cutting. But if we design a guide RNA that there isn't any site with one mismatch, the only sites are going to be with 2, 3, or 4, then we dramatically can eliminate the activity at these sites. The second is to tune the chemistry. You can again tune the chemistry in such a way that you decrease the binding on targets. Since there is a full match, you decrease it to a level that there is still binding, but at the off-target site, since there is a mismatch, you will bring it below a minimum that will decrease the off-target activity. So these are the next level of CRISPR that we are working on.
I must have missed it, but you said that while increasing DNA concentrations, then the editing should favor HDR. Could you be more specific about the concentrations of DNA that favors?
So there are different ways to calculate concentration. many times we actually refer it only as amounts, but when you do electroporation, what is actually happening, and right now at least for these cells, K562 and also hematopoietic stem cells, specifically hematopoietic stem cells, the best way of delivery is electroporation. What is happening is that you're inducing pores in the membrane of the cells, and then the material is getting inside by diffusion. So by increasing the amount of, or the concentration, you can get better delivery of this DNA or even Cas9 protein or mRNA inside the cells. And for this reason, increasing the amount or the concentration is important. And once this material is inside the cells, there are active ways that, or machineries inside the cells that deliver it to the nucleus, whether it's protein, we speak about NLS, but there are also a way for the cell to deliver the DNA and the RNA. So again, since the mechanism is inducing poring, things are getting inside by diffusion, increasing the amount is leading you to a situation where you have more DNA inside the cells, inside the nucleus, available to repair the break by the homologous recombination.
But I imagine if you use more DNA, each electroporation you perform. So
for the K562, the DNA is not so toxic. For primary cells, it's toxic, and we had to find a way to overcome it. And for this reason, we switched to recombinant B6. I hope that this answers your question. If not, we can speak about it also later.
Can you comment about the direction of engineering the protein compared to your direction of modifying the RNA?
Good question. So I didn't mention that, but you can also, in order to increase activity, but even more than that, specificity, you can engineer the Cas9 protein. What is nice about this CRISPR system, which is RNA-guided mechanism, there are two parts, and you can dial each of these parts in order to increase activity and specificity. One is the Cas9 protein, as you mentioned. So what you can do is you can pretty much engineer the protein in such a way that it's going to eliminate unspecific binding. So there are residues on the protein that bind the DNA in an unspecific manner, positive residues. And what you can do is you can try to look for this residue, and there are at least 2 to 3 papers that were published around it that you engineer the protein to remove this residue to decrease the unspecific binding. But the price also with the RNA can be reducing also the on-target activity. So the challenge is again to find a way to decrease the unspecific binding in a way that won't affect the on-target activity. And pretty much what is going on, when you think about transcription factor or mechanism like that, how do you find your target? You need to screen the entire genome in order to find your target. And we still don't understand exactly how the CRISPR system is scanning all the genome until it's finding the target. But one way to do it is by having unspecific binding that will scan and slide across the DNA until it gets to the right site, and over there you're going to get engineering. So there is the balance between the improving the specificity and reducing the on-target activity. Thank you very much.